Morning, everyone. Once again, we are here for another topic of IGCSE Cambridge 0610 Chapter 5, that is enzyme. And we'll go through the updated syllabus of the Cambridge, and we'll try to cover all the sections of the topic. So let's begin. Those who have not subscribed my channel, please you can subscribe. Meanwhile, I'm just going to share the slide and we'll start our discussion. Okay, so here we are with the topic enzyme. So let's discuss some portions which is going to get covered in this section. So here is the syllabus for the enzyme. So we have to describe the enzyme as a catalyst that helps to increase the rate of chemical reaction and is not changed by the reaction. So if you can see in the syllabus, they have changed define to describe. That means they are not going to ask any defined term in the question. Then describe enzyme as a protein involved in metabolic reaction where they functions as a catalyst. Then we have to discuss about enzymes are important in all life. When we say all life, the meaning is the all metabolic reactions is controlled by the enzyme. Then 4, describe enzyme action. So we'll talk about how enzyme and substrate interacts. Then we have an investigation to describe the effect of change in temperature and pH on enzyme activity. And we have also to discuss denaturation. So we'll see what is denaturation. Then we have to explain enzyme action with reference to active site, enzyme substrate complex, substrate and product. Then explain the specificity of the enzyme in terms of complementary shape. So enzymes are specific, how they are, we'll discuss in the topic. Explain the effect of change in the temperature on the enzyme activity in terms of kinetic energy, say shape and fit, frequency of effective collision, and denaturation. And the last one, effect of change in pH on enzyme activity in terms of shape and in terms of fitness. Fitness means whether the enzyme and the substrate is going to bind on the active site or not. So that will be the discussion. So this is all about chapter 5. So let's begin. So in the previous section in chapter 4, we have discussed about biological molecule. So here enzymes are also biological molecule, not in terms of chemical composition, it is in terms of the protein. So enzymes are proteinous, means it is made up of protein. Protein molecules are there. Right. So because there's some glitch in the digital pen, so let me write it again. So enzymes are protein, that's it. So if it comes to define the term enzyme, and the question is of 3 marks, so try to mention the biocatalyst, they speed up the reaction and without being used up in the reaction. So you can define the term. So this is definition, that enzymes are biocatalysts that speed up the rate of chemical reaction without being changed or used up in the reaction. So it's not going to be changed and it is not going to be used up also. Means you will get your enzyme back at the end of the reaction like this. Enzyme combined with substrate, you will get enzyme substrate complex plus enzyme plus product. Okay, so we can say enzyme plus substrate gives enzyme product, right? And we'll get enzyme substrate complex, and then we'll get product and enzyme. So we'll show you in the next diagram how the reaction go on. Okay, so next point says it is made up of protein. So yes, we know that all enzymes are made up of protein, but all proteins are not enzyme. So we have to understand this very important thing. Why we are calling them as a biological catalyst? What is the meaning behind this? Because it is found in the living cell. That is why we are calling them biological catalyst. And why it is necessary? Because it's maintain the chemical reaction in the cell, all metabolic reaction. Example, digestion. So in this slide, we have end up with some key term, like first is catalyst, second is enzyme, third is substrate, four is end product. So what is catalyst? Catalysts are the substance that speed up a chemical reaction is not changed by the reaction. That's enzyme. Protein that function as a biological catalyst. Substrate means enzyme on which it is going to act. And the end product is the result of the reaction. So in this slide, I have already given the how enzyme combined with the substrate to make a complex. This is the complex. And after that, product is formed. And the shape of substrate and product is not going to match. You can see here. In this section, I have drawn the enzyme, and this is the active side, which is V shape. And we have two substrate. So A cannot bind here. B can bind because B has a shape which can fit in the active site of this enzyme, and the complex is formed. And see the product. Product is totally different from this triangle. So this is triangle, this is square, right? So shape is totally different. And this is the enzyme we have got back without being used up and without being changed in the shape. This slides talk about the specificity of the enzyme. Means they are highly specific. So you can see here, this is the substrate, this is the enzyme. Right, we have a two enzyme. So this substrate cannot bind here, but it can bind here. So they have a complementary shape. Complementary means exact same shape, same opposite shape, so that they can fit in the active site. The biocatalyst is unchanged by the reaction. So as I said, the enzymes will be used up again and again in the reaction because it is unchanged. It is not being changed. It is unchanged. So you will get your enzyme back so that it will act on another substrate. So enzymes are specific to one particular substrate. As I said, it is highly unique. As the enzyme active site is complementary to the shape of the substrate molecule, that is why they can fit into the active site of the enzyme. And the product is the shape of the product is totally different from the shape of the enzyme. So enzyme's active site. So like this to the substrate. So sorry. So this enzyme the substrate and the product shape is totally different, as we have seen in the previous diagram. So that's enzyme substrate specificity. Okay, so this is quite complicated diagram. I can see there are so many things. So let me show you one by one. So first, how substrate act on the enzyme. So you can see here is number one, where here it is. So where the substrate is going to bind here, bind to the enzyme, and after binding, they have formed a complex. And after, once the complex is formed, enzyme substrate reaction starts, product is formed, and then enzymes is now free, so that it can act on another substrate. And in this way, the reaction will keep on going. So enzyme substrate interaction is like a lock and key. Like for every lock, there is a specific key. In the same way, for every substrate, there is a specific enzyme. It's not like any enzyme will do the reaction. So they are highly specific. Enzymes. So as we know that enzyme is a protein, right? So they must have a 3D shape, and this shape defines function. So if how the shape is 3D shape is maintained by some folding in the protein that makes active form, and that where that activity takes place on enzyme, that's called active site. So active site is the shape fits to the substrate shape like this. You can see here in this particular diagram, this is the active site. You can see here, and this is the substrate which is bind to the active site through lock and key model. And this also defines specificity. Like see here, for every key there is a specific lock, or for every lock there is a specific key. So this lock and key method describes the fitness, how fit substrate and enzymes are. So, so the question may be asked in terms of how enzyme action takes place, what is enzyme specificity. So how to frame the answer? There are 6 possible points. You can frame your answer in these highlighted specific keywords. Enzymes are specific to one particular substrate, always. That's specificity. Then we have that active site where the substrate is going to bind due to the complementary shape. Why this is happening? Just because of the specific 3D shape of the protein, of the enzyme. And we have just now seen lock and key hypothesis, where enzymes combines with the substrate in a lock and key method. So when the substrate moves to the enzyme's active site, they will form a complex which is called enzyme substrate complex. And after that, product is produced and product is released. Right. And then after that, this product cannot go bind again because it is the product. So enzyme will be ready to bind with another substrate. So this is what we have seen here, that this is the correct enzyme. This is correct substrate. So they can bind here like this in the 2 diagram you can see. And the products are formed, which is quite different from the substrate. How enzyme works? If the question asks how enzyme works and the questions are 4 mark, so you can frame your answer like this: enzyme and substrate randomly move in the solution. When an enzyme and its complementary substrate randomly collides, so actually the reaction takes place due to collision of the molecules. So here also enzyme and substrates are molecules, they are going to collide, and when the collision will be effective, then enzyme substrate complex will be formed, and then product is produced, and then after that enzyme is released, which is unchanged, means it is not being used up. Now we'll talk about investigation uh, mm, part of the topic, where we are going to see the effect of temperature on amylase. Amylase is an enzyme. It's a digestive enzyme, and they digest carbohydrate. So, okay, so let's see what we are doing in the experiment. So starch solution is heated to the set temperature. Iodine is added well to the spotting tile. Amylase is added to the starch solution and mixed well. Every minute, drop of solutions are added to the new well of iodine solution. This is continued until the iodine stop turning blue-black. That means we are confirming that there is no starch left in the solution, as the amylase has broken down all the starch into glucose. Now, time taken for the reaction to be completed is recorded. So we have to keep on checking how much time the solution has taken to stop turning blue-black. Experiment is repeated at different temperatures. So like different temperature means you can take 0 degree, 20 degree, 30 degree, then 40, 50, 60 degree, like this. So, or you can take 0 degree, 30 degree, 60 degree. This is also possible. So two range I have given. So you can say that at 0 degree there is no activity. At 30 degree it's optimum temperature, room temperature. So enzyme will best work. 60 enzyme temperature is high, so enzymes got denatured. Right? We'll talk about what is denaturation in the later stage. Let's go to the next section. So this is a pictorial representation of how the experiment has been done. So this is your sample, starch solution, the blue color. Thermometer is kept to check on the temperature of the solution. And this investigation is done at 30 degree. Second, we have spotting tiles. So there are iodine added to the spotting tiles, like this. Then amylase drop is taken, and we are going to put the amylase starch solution at 30. We'll put it on the tiles, and the timer will start. So as the time go on, then you will find that some of the uh, starch has been converted into blue-black. So we have to record the time. So you can see here at 20 degrees Celsius, means at 9 minutes, this much reaction has been done. At 40 minutes was this much, and at 60, no digestion, no color change. So that means 60, at 60, the enzyme was not working. It's a high temperature. At 40, it was working. 20, it was okay working. I mean, not that much great reaction, but it was slightly working. So 40 degree was the best. In the same way, you can also check the effect of pH on the enzyme amylase, and we have to do the same kind of investigation. So let's see how to do this. So first we have taken iodine added with spotting tiles, same type of reaction. Here we have taken two solution. First we have taken amylase plus buffer to test tube, mix them, add starch solution, and then you have to start the stopwatch. So this is the starch solution, put it in the buffer solution, and then you have a 2 centimeter cube amylase solution. Then timer will start. Right, timer starts. Then after 10 seconds, add a drop of solution to iodine in the spotting tile, like this. So at pH 7, the 60 seconds for reaction to complete. So that means amylase enzyme works in best in neutral pH. Right, so neutral pH means they are effective in pH 7. So see here, when pH was 5, means slightly acidic towards, it takes 80 seconds. When pH was 9, it was taking 90 seconds, and when pH was 14, there was no reaction. So you can say that amylase get denatured in the pH 14, and pH 5 let take, if see 5 is slightly near to the 7, right? Whereas 9 also, but 9 takes, as the experimental result says, that 9 takes 90 seconds to do the reaction, right? Here's 80 seconds pH 5. So based on this, you can repeat the steps once again to find out the more reliable data. Okay, so let's see the effect of temperature on the enzyme activity. So see, just now we have seen that as the temperature is increasing, enzyme is not working. So we need the best temperature. That means the optimum temperature. So optimum temperature is the room temperature. Generally, we can say 40 to 40. Right. So enzymes are protein and have a specific shape. We know that, and the shape is maintained by the bonds present in the protein in the enzyme, and to maintain the active site so that substrate will fit and the reaction will go on. But sometime when the temperature is not optimum, then the bond starts breaking. That means they will break the bond and the enzyme will get denatured. Denatured means enzyme has lost its specific structure. Shape has been lost. And denaturation is generally irreversible. You can't get your enzyme back. So once shape is distorted, the activity will completely stop, or the rate will be slow. So what are the conditions? So at low temperature, enzymes are inactive. They are not denatured, they are inactive. Optimum temperature, enzymes are active. And at high temperature, enzymes are denatured. So that we have to know. So as you can see here in this particular diagram, so this is the normal structure of the enzyme and this is the substrate. So when temperature is increased, you can see the bonding order of the enzyme shape. See the active site shape. So in this, as the temperature changes, shape of active site also changes. So this is very important. Here you can see in this particular diagram. So A, when the temperature was low, it was inactive. Activity is very slow. You can see here. This is the rate of activity. This is y-axis. This is x-axis having temperature. So as the temperature increases, you can see the slope is going up, right, like this. So that means as the temperature is increasing, the rate of reaction is also increasing, you can see. And the best temperature at which enzyme was fully working was this point, where the activity is high, you can see. And beyond this point, like suppose we are here at 40, and after that enzyme is increasing, the temperature is increasing 50, 60, right? So enzymes getting denatured, right? So C is the point where high temperature is there and enzyme got denatured. Enzyme and pH, yes, enzyme and pH have again a very direct connection. So as we have seen that if pH is also not maintained, enzyme activity is going to disturb. So optimum pH for the most of the enzyme is 7, because our body temperature is nearby room temperature, and the neutral pH is 7, which is the best condition for enzyme to work. But some are produced in acidic conditions, some are in the optimum pH, some are in the high uh, pH. Like you can see in stomach, we have acidic condition. We have an enzyme called pepsin, which works in low pH. Some are working in duodenum, they have a higher pH, like 8 or 9, means slightly basic. So if the pH is too high or too low, the bond, again, the bond present in the protein molecules will change. So as we know, the bond order is changing, that means shape will change. And when the shape will change, substrate is no longer going to fit. So again, if you're moving away from the optimum pH, it will cause denaturation to the enzyme. So you can see here, this is the enzyme. See the shape, active site shape, but after pH has been changed, shape has changed. See this one, active site is changed. So now substrate cannot bind. This graph shows the same thing, like for generally for optimum pH is 7 or 8. So the graph here shows high. So optimum pH is this one. At low pH and at high pH, enzyme activities is started because enzyme is not stable. Enzyme is not in a functional form, right? So this is all about the IGCSE biology enzyme topic. Hope you have understood all the concepts here. If there's any doubt, you can write to me and we'll get back to you. Thank you, everyone.